All right. Hi, everyone. I'm Rama, and I'm faculty in the Department of Biology at Ashoka University. My main area is human immunology. And today's topic, I admit, was more as a clickbait, just for attention. So the topic is chicken or egg vaccines and immunology. And this is just a brief outline. It's not a sequence, but just a brief outline of what the topics I'll be touching upon. We start with a short primer on the immune system, then a historical perspectives of vaccines or immunology, which came first. And then in the end, I'd like to talk about why I'm talking about vaccines and immunology at all. Why I think there's a good time to talk about vaccines and immunology. So at any point, just please feel free to interrupt, and you don't even have to raise your hand or anything. There's few enough of us. Just unmute yourself and ask a question. So at any point you want to, there's a point of discussion or something, just please go ahead. So I'm going to start with a primer on the immune system. So what is the immune system and where is it, right? Broadly, we can think of two types of immunity. One is innate. The word means that with which you're born or that's present naturally. And the other is adaptive or arising as a result of adaptation. So the innate immune system can broadly be thought of as both like physical and chemical, pre-existing physical and chemical barriers that prevent pathogens from entering. And these include, for instance, skin, right? Physical barrier. It provides a physical barrier. There are commensals on the skin, and there's chemical barriers such as low pH and commensal in the vagina, which commensals essentially outcompete the actual pathogenic organisms, and so on. Right? There are some mechanical barriers as well, things that make you sneeze when something goes in, acid in the stomach, and so on. And apart from that, there are cells of the immune system, of the innate immune system. There are macrophages and dendritic cells and so on, which actually work towards triaging the infection initially and educating and either clearing it, or if not, then helping the adaptive immune system later on to make a more tailored response. And apart from this, there are proteins and peptides, complement defenses. And when you think of the adaptive immune system, you can think of it broadly as comprising either of antibodies, or broadly it's referred to as the humoral immune response. You have B cells that differentiate, become plasma cells, which are factories of antibodies. I think given the pandemic, all of us now familiar with terms like antibodies and IgG and T cells and immune memory and so on. That's one of the adaptive immune system. Second arm you can think of as the cell-mediated response, which is where CD4 and CD8 T cells come in, or they're alternately referred to as helper T cells or cytotoxic T cells. Whereas broadly, the humoral immune response recognizes pathogens outside the cell. Usually, broadly, the cytotoxic T cells will recognize pathogens that are inside the cell. For example, viruses. Viruses are mostly, they can infect inside the cell, and those are recognized by T cells. So what is unique about the adaptive immune system? The innate immune system, as I said, it is pre-existing. So it can recognize a limited number of conserved microbial structures. By limited, I don't mean in any way that there are only 2 or 3. It just means that for that organism or for that cell type, the innate immune system does not learn further new patterns. If there were a new pattern to emerge, maybe the innate immune system would not recognize it right away. So it does not adapt. Whereas the adaptive immune system is special because it can recognize any microbial structure, right? So antibodies can recognize any 3D structure in the extracellular environment. T cells can recognize any linear peptide in the intracellular environment. And this is amazing to me, and this is kind of obvious to us also, right? We have new pathogens all the time. There was I think the first There was I think in recent times, there's been an Ebola outbreak in 2015 that was discovered in 1976. SARS-CoV-2, living through a pandemic right now, that is a new pathogen. But it's not that your immune system didn't recognize it. People did make an immune response. Most of them survived and actually made a protective immune response. So this is just to highlight that the adaptive immunity can recognize any microbial structure. So where is all this? Where is the immune system? We've just spoken broadly about what the immune system is. Where is all this, right? You can think of the immune system or immune organs, existing in the lymphoid organs. There's the primary lymphoid organs, bone marrow and thymus. This is the area where the immune system learns things. They are trained, cells are trained. And the secondary lymphoid organs, so you have the lymph nodes. That's what the doctor checks if you go sometimes when you're sick and you go. That's what they check for lymph node inflammation. So you have lymph nodes, tonsil spleen, Peyer's patches, and the very big mucosa-associated lymphoid tissue. And this is the area where the immune system actually works. And one thing that I've not pointed out here is the lymph, right? The lymphatics, or the lymphatic system, which is right here. There's no text here, but hopefully you can see on the slide. The lymphatic vesicles, which form the traffic way of all these cells to move around from where they're learning to where they're supposed to do their job. Now, what does the immune system look like to me in my world is that we look at immune memory and or immune memory or immune responses to, my interest is immune responses to vaccines or to viral infections. And the easiest thing to track is blood. So this is what blood looks like after it undergoes density gradient centrifugation. Most of it is plasma, which has water, proteins, and so on. And this is where the antibodies reside. And then there's the red blood cells, which we don't like to look at. But the cells of my interest are, because I'm a T-cell biologist, is what is called the buffy coat, which has both platelets and leukocytes. And when you do a density gradient centrifugation, this is shown on the right. So these are the cells that we isolate and then characterize further. Now, one thing you'll appreciate is because of the organs being so dispersed in the body, it's not one organ. And also because the conduits, the traffic ways that the cells use are blood and lymph. One thing that you can appreciate is that the immune system is everywhere, right? It is everywhere. It is not one single organ, and then that makes it a little bit difficult to study. And apart from that, the other complication is that there are many different cell types in the immune system, and not just many different cell types. They are capable of differentiating. They exist in different states depending on what pathogen they are seeing, what they are doing. So their location and their function is very context specific. And also they use different mechanisms and means to perform their functions, to protect the body from disease. And this is just a bunch of cytokines and all listed here to illustrate that. So this is just to say that the immune system is complex, and any statement, so which is why any statement that people make these days regarding the vaccine or its efficacy is not, it's a good scientist will never say this is 100%, right? Because they know that there are all these complexities and there are always exceptions that can occur. So this was just my way of saying that the immune system is complex, and I think all the information or misinformation that we're seeing these days needs to be looked at with scientific rigor and evidence-based medicines to understand the data, and that is what is lacking these days. Like I think before the pandemic it was fine. I was just doing my research, and not these terms like antibodies and memory and vaccine efficacy were not so much news among the common public, but now you have everybody talking about them. And then, of course, they've used WhatsApp University, and it's very hard to counter. So I think it's important to educate, undergrads especially, about at least the basics of the immune system, and especially develop a line of critical thinking. All right, so how do we all learn about this? And this is where I'll touch upon about the immune system in vaccines, chicken or egg situation. Some historical milestones are the earliest mentions of infectious diseases occurs in the year 900 by Rhazes, a Persian physician. And the nice thing was he actually distinguished between smallpox and measles, which was very seminal. Very new for the time. Then 1500s, there's an account of inoculation of smallpox in China. And really the history of vaccination, I think, is in many ways the history of the smallpox and smallpox vaccine. 1700s, there's Lady Mary Montagu discovered that, she noted that in the Ottoman Empire, she'd spent some time there, that variolation or taking dried pustules from people who are recovering for smallpox and blowing it in the nose or wounds of other people actually protected them. Of course, this is not 100% safe experiment or a 100% foolproof experiment to do, but this is like a big finding at the time. She brought that technique to England and to the West, and that's when they realized that this could be done. And the concept that you can be protected from an infection. Then there was a Jenner's demonstration of the smallpox vaccine, and many more. So one of the important findings was that immune memory exists, and of course they didn't call it memory then. This was just an observation made by Peter Penham, who observed a measles epidemic in the Faroe Islands, which was very isolated at the time. And the observation that he made was that people who had been infected in a previous epidemic many decades ago were the ones who were protected. So if you just move on ahead, it was say 1877 or so, since when people started talking about not just how to protect from disease, but what is protecting from disease. So all these, for instance, the description of mast cells of being a separate cell type was made by Paul Ehrlich in 1877. Then Meshnikov discovered phagocytosis. There's a concept of therapeutic vaccination introduced by Pasteur. And then just jumping ahead to the 1950s, 1960s, clone selection theory, I'm not touching upon it, but it was a very, it is the cornerstone of modern immunology, I feel. Just the finding led to a lot of other things. So the point I'm trying to make is that the earliest findings were all based on some sort of vaccination or protection from disease, or vaccines themselves. The whole goal was to either protect from disease through vaccination, vaccine being some sort of attenuated or weak constraint of the disease agent. And it wasn't until much later that people started thinking about how and mechanism and actually making findings. And of course, all this is to do with technical advances, conceptual advances as well. Conceptual advances meaning the germ theory being accepted that pathogens can actually cause disease and so on. And this is kind of the reflected even in the Nobel Prizes. Okay, I'm going to come to that a little bit later. But the first Nobel Prize in Physiology and Medicine was actually for you can call it like passive immunization. It was given to Emil von Behring, said that serum taken from animals immunized against diphtheria can protect unvaccinated animals. And at the time, they didn't really know much about the structure of antibody or how it's being processed and so on. And surprisingly, although vaccines have had a huge impact on human health and life and on teaching immunology, The only Nobel Prize for a vaccine, I'm sorry, the name's wrong here. It's actually, this is Max Tyler. It's Max Tyler who got a Nobel Prize for a vaccine for his discoveries concerning yellow fever and how to combat it. So I hope I've sort of convinced you that, at least in the early years, it is vaccines that came first, or vaccine knowledge, or the motivation was to make a vaccine, and that led to findings in immunology. Here's a vaccine, how does it work? And that led to the field of immunology. And I will now go on to some modern times. This is just a brief of flow cytometry, the technique of flow cytometry. The point that this is just to illustrate that a technique that's used a lot in immunology. Okay, so I can't give a talk without bringing up flow cytometry. So essentially, flow cytometry is a technique where you take a mixture of cells in suspension, then you stain them with fluorochrome tagged antibodies to make them visible. Then you do then the instrument, the flow cytometer, then focuses the cells in a single file. There's hydrodynamic focusing, and then there's signal based on these tagged antibodies, and then you get output. So what you see is just like a simple dot plot showing marker for CD for helper T cells on the y-axis and cytotoxic T cells on the x-axis. So using flow cytometry, this is a former colleague's work where he looked at immune responses to influenza vaccination. And so if you can just look at these plots, you can see some phenotypic markers that they used to identify antibody secreting cells in the blood. So just after vaccination, you see this huge population, transient population, which goes down. The rest of the paper, they did show that this actually were influenza-specific cells. These are cells secreting influenza-specific antibodies. I'm using this slide to illustrate two things, okay? The first one is that vaccines, again, the influenza vaccine, again, educated immunology. So these markers and this population and the phenotype of this population, what antibody secreting cells look like, and a generic set of markers that can be used was identified using this vaccine. The second point I'd like to illustrate is if I can draw your attention to the graphs below. This, especially, I'm pointing to B here, where it shows influenza-specific IgG antibody secreting cells per million PBMCs, which is, peripheral blood mononuclear cells. What you can see is, the kinetics is very, very predictable among these 10, 12 donors. And this is, after this, there have been many, many studies done, and the kinetics is quite, it's very predictable. So if you use these 3 time points, you see a peak in a very narrow range of time. The second thing to appreciate is that, look at the variation, right? The number of cells varies a lot among individuals. This is part of the reason why it's very difficult to predict, or it's very difficult. This variability is the reason why I think the general public, maybe it's hard to communicate to them that vaccines work, but even a great vaccine might not work 100% of the time. But that's not a bad thing. This kind of variability is part of biology. Just another example, this is looking at cytotoxic T cells after yellow fever vaccination, showing an example of what antigen-specific cells look like using a reagent called MHC tetramers. Won't go into that now, but again, just to say that this is a similar graph. Kinetics, each line is one donor, and this is time past yellow fever vaccination. And you can see that the kinetics are remarkably predictable. There's a certain burst of proliferation. You see a peak around day 14 after vaccination. And the second point is that it is quite variable in magnitude, right? So there is variability, but the one thing we do know about at least the yellow fever vaccine is that it's very successful, and it it's one of the most successful vaccines ever, 98, 99% seroconversion and so on, right? So again, this is another example of a vaccine that has taught immunology. So like both, I'm just sort of I'm using this plot to illustrate the kind of questions you can ask, especially in humans. Because humans, you're very limited by the kind of samples you can get. If you wanted to track the kinetics of or the immune response to a pathogen, your local immunologist is not the first person you think of when you get sick, right? So it's very hard to perform longitudinal analysis or get cells at different time points or characterize cells or actually know much about the immune system with or using, say, a pathogenic, using a pathogen. And this is where vaccines help, because they're safe. They are, you know exactly when they're given. And you can ask very simple questions, fundamental questions in humans, like clonal expansion. What is the time? We had used the smallpox and yellow fever vaccine to ask questions like, okay, so how do we identify effector T cells? And we found these four generic markers that can be used. Then you can ask questions about immune memory and so on using vaccines. So here's a great vaccine, elicits great memory. What does that memory look like, right? So some modern milestones in immunology in the 80s and 90s had the TCR, the T cell receptor structure discovered. Then James Allison pioneered research into CD28, another one called CTLA4. Tasuku Honjo discovered this molecule called PD-1. And not to go into a lot of detail, I just like to say that the T cell receptor can be thought of as the ignition switch of a T cell. So engaging the T cell receptors is the one that senses that there's a pathogen or there's an infected cell. And by sensing that, it can then make more it can undergo clonal expansion, basically make an army to remove those infected cells, right? So if you think of the TCR as an ignition switch, CD28 becomes the accelerator. So CD28 is another surface molecule, and engaging it along with TCR is very important for the T cell to really go ahead and expand. And using the same analogy, PD-1 and CTLA4 can then be thought of as brakes of the immune system, because you don't want that response to go on forever. It can actually be detrimental in the long term, right? So connected to this, these findings, they've just not fundamental findings, they've actually been used. So fundamental immunology findings have been used for human health. Just showing you two examples here. The first one is cancer T cells, cancer vaccines, and T cell therapy. CAR T cells were actually FDA approved in around 2011. And then you must have heard the term immunotherapy, and that has come into play because of the idea that if you reverse the brakes, PD-1 and CTLA4, and this is brakes are turned on, especially in situations of chronic infection like HIV or in cancer, and treatment or reversing brakes through treatment with anti-PD-1, anti-CTLA4 can actually be used for therapy, and it has been used successfully for many cancers since 2014 when it was first licensed, right? So I am going to say that, at least in modern times, or more recently, it is immunology that has educated vaccines. So the answer to the question is, I think it has changed depending on the times. If somebody writes a history of our time, like hundreds of years later, the answer will probably be this: that it's more that learning immunology is important to design vaccines. But of course, like the chicken and egg situation, they both feed into each other. They both educate each other. Vaccines, we learn immunology through vaccines, and we design better vaccines by learning immunology. Just like I just illustrated, a fundamental immunology finding led to a great cancer therapy, which is immunotherapy. And this is just to tell you about how great the finding about the PD-1 finding. This was the most recent, or not the most recent, but fairly recent. This is 2018 Nobel Prize in Physiology and Medicine was for the discovery of cancer therapy by inhibition of negative immune regulation. James Allison for CTLA4, Tasuku for PD-1. All right, I think. So I'll just end very quickly about why I'm talking about vaccines and immunology. So I actually just found out yesterday that you can get fake certificates of vaccines, and then also about various people not getting vaccinated. This is in the context of the COVID-19 vaccine because of various reasons. And so this was a little, this still shocks me. And I think there's a lot of misinformation and disinformation about this. And at least we can counter the misinformation by educating ourselves better. Even if you're not interested in immunology, given the pandemic, I think we should encourage ourselves and people around us to just think critically. But how do you think critically? Because the news, these are our sources of information, right? The social media, there's so and so, friends of friend, oh, they got the vaccine and then they lost all their hair or some such thing. Personal experience from the past, dated information, maybe something I learned in school like 30 years back, or it's tradition, there's peers or something. Okay, and then there's heavy duty stuff like scientific journals, right? So how do you, if you're not an expert, how do you do this? The first thing is to just think and ask the right questions. Don't assume, and ask the right questions. Listen to a lot of talks by experts, not again social media or forwards, and learn to think critically. And then also educate yourself. And I just put some resources for learning and teaching immunology here. A lot of them are available freely, and I really appreciate it, because science communication to a general audience is difficult, and then these make it very easy, and you can just just go here and explore and kind of educate yourself about what is true, what is possible, and what is not. All right. Thank you.
You showed that data that you showed. Which SARS virus was it? I mean, it's a 2008 paper. Which SARS, when they showed
no.
Rama
Data, I only showed two pieces of data. One is influenza vaccine and the other is a yellow fever vaccine. That's it.
Right, but it said, I think it said Rama et al.
Yeah.
And
they may have done work in SARS-CoV-2 recently, but that's 2008, and that's influenza vaccine. Yeah. SARS, I think they are doing a lot of, I didn't include SARS deliberately, partly because there are so many papers that they are rubbish. It's very hard to sort what is rubbish and what is not. I'm just, it's so hard to keep up with all the literature. Like there's one paper which sort of concluded using some sort of genomic analysis that plasma cells become T cells or some such thing. It was not and without any evidence, and they, so yeah. So yeah, I didn't think it was wise to include SARS. There's too much literature. Yeah.
I saw that there was a large. Oh, Shivani, please go ahead. Yeah.
Did you want to finish what you were saying, Lakshman?
Oh, I can come back to it. But I mean, Rama, I saw a large increase in the number of papers being put out. They are not peer reviewed yet, but they're on the BioArchive. I don't know if this was meant to sort of expedite discovery in the days of COVID, but there have been a sharp uptick in the number of papers on influenza and such. Have you?
Yeah, see, I have no problem with BioArchive or MedArchive. I think it's a great idea to, and these are, and they openly, they say they're not peer reviewed. And I think it started out as a way to publish your paper and then get feedback. It's kind of like me giving you my paper to read, saying, listen, is it written well? Do you have any, any controls I should include, and that kind of thing, which is great. No, the papers I was talking about are actually, they are published papers. They are peer reviewed and published, but obviously there's been a race to publish first. So, those are the papers I was talking about. And then now you have to go back, and some have been retracted also. Some have been retracted like within days of being published. So yeah, those are the ones. But I think MetaArchive, BioArchive are good because they say that they're not peer reviewed. And you can immediately, if there's something wrong, then you can openly, there's a space to comment. You don't have to go through a long winded route. You can just leave it in the comments, and then I think there's a paper from IIT Delhi about it was about SARS-CoV-2, and they did some sort of genomic comparative analysis with HIV or something, which was pulled out very quickly. But and that was BioArchive. It was not published. No, the papers I was talking about was peer reviewed. Anyway, if you guys think of questions later, then please email me. Both Lakshman and Shivani have my email. So
I had a question. Thanks, Lakshman. I had a question, Rama, and this might be tangential, but with this newest variant that's out there that's causing fewer symptoms, there are again, you said don't go to media, but there are opinions that come from experts that use epidemiological, public health, and evolutionary arguments, both for and against getting infected purposefully to get done with a less deadly virus. Does the field of immunology have a role to play in informing opinions in this aspect, or does it just stay in the realm of public health, I guess?
Yeah. See, I'm sorry, my intent was not to say that don't go to the media. My intent is, often I tend to what you're listening to is usually, it's not like an entire talk. It's an expert opinion, and it's either edited out, or it is very very short answers meant for the general public, which is fine. But what I'm saying is, you should just go there, but don't question it. Okay, don't believe it completely. I tend to be very easily led. Okay, when I read a paper, I say, oh, great. I never think critically till there's a journal club and then someone says they didn't do this control, or the interpretation is wrong or something. That's all I meant. The role of immunology. So one thought I have heard, one school of thought is, this deliberate infection. It was to do with, frankly, I don't know. I just don't, I don't know, like one of the things that has been going on is, other thing I heard is that the Omicron variant is God's vaccine, because everyone will be infected and therefore protected. The other school of thought I've heard is it's actually bad to get vaccinated, because then you have immune escape. So that provides pressure for the virus to escape. As in, if you make antibodies, there are viruses that are not selected or neutralized, and then those it actually acts, the antibodies or the T cell response actually has a selection pressure, which is worse. So you should not get vaccinated. That's the other. The point is that it's not that straightforward, and we need a lot more sequencing data, viral sequencing data. We need a lot more, especially in India. That's one thing that bothers me. We don't have easy access to viral sequencing data, antibody sequencing, antibody repertoire analysis, which we are not doing to know about this. It's a very roundabout way of saying I don't know, but realistically, that is it. I think the answer is somewhere in the middle. Yeah.
Thank you.
I had a question. Thank you for the talk, Professor. My question is about what's the difference between vaccines that can prevent disease and vaccines that are just protecting against severe disease? Like, what's happening with respect to the body's response, that in one case you have lifelong immunity, while in the other you can still get infected, like with COVID vaccines? Yeah.
Okay, that's a very deep question. I think the question is, why do some vaccines elicit long-lived immune memory and some do not, right? that is a question that, I plan to, I think, run my all the projects in my lab on. It's a very interesting question. See, the one common thing, sort of broadly, roughly, is that anything that's an acute infection, so like the yellow fever or yellow fever vaccine, chickenpox, measles, these elicit long-term memory. Something about virus being, something about the nature of those viruses, that when they are, when they come up and then they subside, the immune system is able to clear them in a certain time frame. You get long-term memory, right? Now, what that memory comprises of is, so one way to address the question is you can read, there's a paper by Hamilton et al. So where they examine this, you can examine this in humans again with the vaccine, right? The smallpox vaccine. It's been around for a long time. Or they started vaccinating in the 70s, in the late 70s. But so what they did was they asked this question that you're asking, part of it. What is it? What is immunological memory? And what they found was that essentially you found both memory T cells and memory B cells. But what was longer lasting was the memory, was the B cell memory, the antibody part of it, the cells that make antibody. Okay? These antibodies were longer lasting than T cells in most people, right? So first, you have a good chance if there's an acute viral infection, which is not too, widespread over the body, and then you get one of the characteristics of long-lived memory will be that probably antibody, in some cases T cells. So that's one specific answer I can give with one vaccine. The rest, in general, if you have a chronic situation like HIV, long-lasting virus can hide, it's latent, or you have a situation like cancer, which is again a chronic, you can treat it like a chronic virus. It leads to something called T cell exhaustion, and does not lead to memory. Okay, so bottom line, it depends on it depends on the virus, the nature of the virus, and it also depends on the actual, and it depends on various components of immune response. But it's a very deep question, and it will take like a lifetime to talk about these things. There's no one size fits all. There's no one size fits all for vaccines and memory in general.
Yeah, if I can just follow up.
Yeah.
Is antibody count like the only way to measure immunity? Like, is that the only marker we have?
I wouldn't call it marker. We're talking about antibody levels. The answer is no. That's not the only way, because you have T cells, right? You have T cells that are protective, and this idea that antibodies can provide sterilizing immunity doesn't always happen. Okay, sterilizing meaning the minute the virus enters the body, there's pre-existing antibody, it'll just bind the virus and prevent the virus from infecting more cells, right? Sterilizing immunity. You can have antibody acting through other, and that's what people look at when they look at neutralizing antibodies and everything. But anyway, I'll keep it short. The answer is no, antibody is not the only measure. But see, all these things, no, that's a huge question. What is the correlate of protection? Correlate. So this is very nicely explained in a book called Vaccines by Plotkin and Orenstein. So a lot of these are population-based studies, historical studies saying so many people are vaccinated. What is the minimum titer above which they were exposed to the virus or lived in an endemic region but did not get the disease, right? So when people say the correlative protection is antibody, it doesn't mean that only antibody is the only marker or the only reason why they're protected. Okay, I hope you notice the difference.
Yeah. Thank you.
It's just T cells are harder to measure.
Okay.
Antibody, you can have one assay that you can do for everybody, but specific T cells, I would have to tailor it according to the MHC haplotype of the person. So they're harder to measure.
Okay.
May I ask a question?
Yeah.
Sorry. Thank you. I apologize. I ran from one meeting to another, so you may well have answered this in the first half of your talk. But I one of the things that interests me the most is the history of biology. And I was really happy to see the time that you spent sharing this. And I'm curious, what's interesting about the first decade of the Nobel Prizes is the attention paid to both immunology and germ theory, in that over half the prizes went to these at that point. And you point to Ehrlich's magic bullet and the like. But then you have this scattershot. You've got McFarlane Burnett, and then you've got what happened in 2018. And like you have, germ theory stays on, and there's this great interest. But is there a moment in the history of the 20th century which marks the high noon of immunology? And when would that be, and why? Or is it that, like with germ theory, you've had this essentially going gradually across and casting its light, so to speak, rather than shadow across it?
I think thing is, when you read about the history of immunology, I'm looking at it. So everything is condensed into just one slide or one set of findings for me, right? But I think my favorite finding, or what I think was very important, was Panem's observation that I would say that is a turning point. And then, Pasteur did a lot, and I think some people would consider him the father of immunology. But I'll come to that. Okay, so Panem, when he said that, okay, it was just observation, right? Going and observing, it was a natural experiment. Measles hadn't occurred in the Faroe Islands for a long time. So I think that led to a lot of thought and reflection about immune memory, and that you can. So that for me. Pasteur did a lot, and they actually were, they thought differently. Their thinking was very creative, and they were really very driven. But sometimes, I just question the ethical, the ethics of the experiments. Like, he essentially infected they were a young boy. He gave a vaccine that hadn't been tested.
Of course, Judah might say, of course, yeah.
Yeah. So that kind of colors things for me a little bit. I'm sure they're great scientists, but somehow I just can't, help not thinking about it. Yeah. Great question. Thanks.
Thank you very much. Thank you.
Fascinating talk. Again, sorry I couldn't join earlier. Maybe you answered this. But coming back to this medArchive and BioArchive, so is it just me, or like I've noticed that just in the recent, since COVID, the number of these pre-reviewed articles getting cited in the
pre-publication
getting cited in the regular journalism has kind of increased tremendously. So how do you see that affecting how immunology is communicated to the general the wider public? Because of course these studies are not peer reviewed, and also I kind of find it troubling that there is not a whole lot of studies in peer J. So I think for other more open access journals, I guess there's pressure to publish in top journals, and also you'd have thought that those other alternative venues would have been the choice to go to, but it's kind of a weird dynamic playing out.
So I think you're right in that it's during the pandemic that the BioArchive, sites like BioArchive, medArchive have seen a lot more papers than before. I think the reason is twofold. One is that it's just timing, So I remember like a few years back, like before, I think this was maybe 6-7 years back, I told my former boss, I was working with him as a postdoc, and I said, listen, let's send this paper to, we were kind of arguing about something, about interpretation or something. And I said, let's put it in BioArchive and see how it goes. You can share it with the other scientists. Let's see how people give. My whole point was that we would get some feedback. And he was like, no. And, so he didn't know about it. And then he also was against it. What is this kind of, it's a paper, but it's not a paper, and that kind of thing. But then now he himself is, he has papers coming out in BioArchive. So part of it is awareness, part of it is a race. You do want to publish first. I understand in the UK and all now, even for PhD requirements, they accept BioArchive papers. Part of it is and also, the peer review system is not perfect. There have been flaws, as in, that's the reason you have retracted papers. The pros I see as far as immunology or human immunology is concerned with these preprint servers is that one, of course, access to information. We have, in general, even with the pandemic, there's access to information, very quick access to information. Nature papers, science papers, cell papers can take a long time to turn around, especially if they go about asking, mechanism-based questions, or they give you some experiments, like especially human experiments. How do you do them? Even mouse experiments, for that matter, it's at least 6 months. So I think it's access to information, which is great. Yeah, the common point between whatever journal it is I think you all have to learn to think critically and question. Don't let the authors lead. Don't go by the author's pedigree or how many papers they've published, and learn critical thinking. And that's something even I still struggle with. I still try and learn. So yeah, I don't see a negative. Let me put it this way. I do not see a negative with having BioArchive or MedArchive. I think it's a good thing. And if you read those papers and if you find some sort of mistake or comment, or you have questions, you should certainly post there. You should ask the author. You should call out the authors. So I think it's a great forum. No negatives.
Thank you.
Thank you, Rama. And if there are no more questions, thank you again for a nice talk.
Yeah.
Thank you again
so
for coming
much.
by. Yeah.
Thank you for inviting
so
me.
much.
Yeah, thanks. Bye.
Yeah.
Bye-bye. Thank you so much.